Biomedical Engineering and Ophthalmology at the University of Texas Southwestern Medical Center in Dallas. Dr. Rizwan received a PhD in Biomedical Engineering from the National University of Singapore in 2017 and completed his postdoctoral training at the University of Toronto with Dr. Molly Shoichet. And Rizwan research group is currently focusing on engineering different type of hydrogel-based biomaterial for tissue regeneration. The overarching goal of his research program at UTSW is to leverage the adaptable nature of hydrogel to modulate cell functions, create transplantable tissues, and precisely control the delivery of biotherapeutics for in situ regeneration. Dr. Rizwan is a recipient of prestigious Singapore International Graduate Award for Doctor Research, and recently he has awarded two big grants like NIH R01 and R35 for the current project. So he's a very good in designing different type of hydrogel, especially like ocular regeneration. And he's one of my fellow friends and a partner in crime. Some of the research work usually we discuss. And thank you, Rizwan, for coming here. And we are excited to have you here. And the floor is yours.
Thank you, Johnson, and thank you for the invitation. Thank you for the nice introduction. This really feels great to be here. This is my first time. Heard a lot about Terasaki. Just the environment is awesome. Like this feels like an open space where I'm presenting, and this is the first time I'm doing this. So awesome. So okay, I'll share my research today from our group on basically designing hydrogels to improve tissue regeneration processes. Currently, I'm a faculty at the Department of Biomedical Engineering and UT Southwestern. I started as a faculty in Michigan Tech in 2022 and moved to UT Southwestern last year. This is basically where our lab is. This is a new UT Southwestern / UT Dallas biomedical engineering building. The concept was intriguing, that having a biomedical engineering department which is in a medical center. So that's what basically pulled me towards this place. It really allows us to collaborate closely with the physicians and clinicians at UT Southwestern. Pretty much all the clinical departments are within walking distance from this building, and so that really allows the collaboration. So I currently have a joint appointment in ophthalmology, but we collaborate with hepatology. We also collaborate with neurology departments there. So far, pretty translational research, and there's a lot of focus on the BMB program from the leadership at UT Southwestern, so which is always good. Okay. So as Johnson mentioned, the overarching goal of our research is basically designing hydrogels that have unique properties. We really love these hydrogels because of their tunable nature. These hydrogels, we focus on basically combining organic synthesis approaches with material science to try to create hydrogels that can respond to different stimuli. For example, here you can see this light or 2-photon laser that was used to create or basically conjugate model drug molecules at very precise locations inside this hydrogel. We are also focusing on the release of different growth factors from these molecules. Another big focus in my group is using dynamic cross-linking to create cross-links that can respond to cells when the cells are encapsulated in them. And that will become apparent as I present some of this work. And then also biomaterial induced signaling. So we can use these hydrogels as a template on which basically we can add signaling molecules that can direct the growth of the cells inside the matrices. We use these as a platform to not only study basic cell matrix interactions, but we can also study or apply them for translational applications that include corneal and retinal regeneration, liver tissue engineering, and recently we have been getting into the stroke repair research as well. So through this presentation, basically I'll share mainly two stories. One is on corneal regeneration that is now funded by NEI, the liver tissue engineering program that was previously funded by NIDDK through a grant. And then towards the end, I'll share some new biomaterials that I hope you may not have heard about previously. So the first story, and that may also be something pretty new to you. So the cornea is basically a layered structure, and it is pretty much responsible for the 2/3 of the refractive power of the eye. So The innermost layer of the cornea is a single sheet of endothelial cells called corneal endothelium. What happens that as we age, some patients, and could be multiple reasons, could be linked to genetic reasons, it could be other reasons, but these endothelial cells start to die because of the kind of formation of these microstructures on the basement side of the cornea. Now that is a big problem because that endothelium monolayer is responsible for maintaining the transparency of the cornea. So even if this endothelium layer is damaged, that leads to cloudy cornea, which eventually needs a cornea transplantation. Currently, there is no disease that can stop or halt the progression of the death of these cells. Cornea is one of the most transplanted tissue, and almost 40% of the corneal transplants happen just because only that layer is damaged. So the rest of the cornea is perfect. But because there's no cure, so we have to transplant the whole cornea. So what tissue engineering promises is two different approaches. We can isolate these corneal endothelial cells from the cornea, the human donor cornea. So instead of doing a one cornea to one patient transplantation, what we can do is we can isolate these cells. And this in blue, you can see the endothelium fragments isolated from a human cornea in my lab. We can isolate these cells. We can try to grow these cells, which is pretty challenging actually to grow these cells in the lab. And after the growth of these cells, after the expansion of these cells, we can use this for cell injection therapy into the patients in the anterior chamber with an idea that these cells will populate or attach to the backside of the cornea and will reform the monolayer. So this is already in clinical trials in Japan. So there is a lot of success basically in this process. And they are currently, actually I was in the GRC in cornea, and the people who are working in Japan, they also presented their latest clinical trial results and showed basically pretty awesome results. The key challenge is ensuring that the cells remain mature, which they do not. So that is one of the key challenge in the expansion. The other approach is that instead of cell injection, we can grow a monolayer on a hydrogel film and then transplant that sort of tissue engineered graft in the patients. This is what the cells basically how they look like in the native cornea, but over the course of passages, what happens that when we grow these cells, they start to lose their markers. A key marker that is clinically used is the morphology of these cells. Usually these cells are really nicely polygonal in shape in the native conditions, but as you grow them, their cell circularity decreases, their cell density will decrease, and the cell area will increase, the shape becomes irregular, and this is happening just over 2 passages. So you literally can't even expand these cells for 2 passages. That is one of the key challenges that is still lingering on in this field. For example, this figure shows that within the same area, there are some cells which do not express Z01. They have lost their phenotype, but some cells express nicely this Z01 marker, right? If we take all these cells and inject into patients, the clinical trials show that almost 50%, 56% lower cell density of monolayer is achieved if you are injecting a mixed population of cells, which are both mature and immature cells. So you sort of have to ensure that as you're growing these cells in vitro, you maintain their phenotype for a good therapeutically relevant cell product for injection therapy. And also the cell loss is significantly higher if you're not purifying the cells. Now, during my PhD, I was fortunate to have been working with Singapore Eye Research Institute, which are one of the leaders in corneal endothelial cell graft transplantation. And where we tried to see, and I was in Dr. Evelyn James' lab, and the lab's focus was looking at cell matrix interactions. So we started seeing, okay, can we improve the growth rate of these cells and their markers by having the same plastic surface that everyone uses, but imprinting these nano features or micro features on those surfaces. And we did see that some particular type of features, particularly those which are in 1 micron pillar range, they could increase the Z01 expression at P3 or passage 3. Now, that may not sound great, but considering that these cells are extremely difficult to grow, and if you can maintain the Z01 expression at even just 1 passage number higher, that's already considered pretty unique. And the proliferation rate of these cells was also higher. So these were showing that indeed these cells have mechanotransduction signals that can basically modulate their fate. Now, another thing, I'm talking about 2012, 2014, and during that time, there was a paper from Christy Anteth's lab, which caught my attention. The paper was focused on They coined this term called mechanical memory, which basically means that the cells were able to remember their past events in terms of the substrate on which they were grown. And that was stored as their epigenetic memory in these cells. And they also showed that you can reverse that memory by playing with what kind of matrix you are using to grow these cells. And then I thought that we are growing these cells on topography, and then we want to inject these cells eventually for cell therapy, where the cells may not have topography. So do these cells still remember the effect of topography when we passage them on a flat surface without the topography? And indeed they do. We showed that compared to the control conditions, the cells that were passaged from topography and cultured on unpatterned surface still had high Z1 expression compared to those that were cultured on unpatterned surfaces. So this showed that to some extent, we can improve the therapeutic potential of the cells by growing them on a surface that has these architectures. And then if we transplant these cells, they should be therapeutically more relevant. Now, when I started my lab, I started thinking about whole mechanical memory thing and how that's affecting the corneal endothelial cells and how come no one is paying attention to that. Now, if we think about this, we isolate the cells from human cornea, the patients. So they're coming from a very soft environment. We expand them in plastic, again, exponentially stiffer surface. But for cell therapy, we inject them back into the patients, right? Again, soft surface. And that expansion phase takes about 2 months. How does that expansion changes the properties of these cells? And endothelial to mesenchymal cell transition is the key reason why these cells lose their phenotype, and how that mechanical signal that they receive basically cause the endothelial to mesenchymal cell transition. So we thought, okay, let's compare freshly isolated cells from the cornea and compare them with those cells that have been cultured on plastic for a couple of passages for about 1 month. And indeed, the RNA-seq analysis showed that the native cells are significantly different compared to cultured cells. We further looked into the gene expression analysis of these cells, and we noted that almost 276 genes have almost over 5-fold difference, native versus cultured cells, right? Now, we were more interested in those genes which are implicated in mechanotransduction pathways, because we believe that we can find some answers of how to make these cells therapeutically more relevant by not just focusing on biochemical cues, but by also focusing on their mechanical environment in which these cells are being grown. So when we analyze the differentially expressed genes, the analysis showed that, for example, a lot of those genes that are differentially expressed are part of these cell substrate junctions or focal adhesions, actin binding, tubulin binding, a lot of those things that you would expect to be involved in the mechanotransduction pathways. Okay. So what we are doing now then with all this information? So what we are doing now in our lab is we have a lot of focus on epigenetic changes that happen in these cells. Not just Christy Anset's paper. Over the last 10 to 12 years, multiple labs have dozens of papers showing that the cells that you are culturing for cell therapy, that their substrate has a strong influence on them. And if you want to maintain their therapeutic potential, you need to look at what kind of epigenetic memory these cells have, how those epigenetic markers are changing when you are culturing on plastic, and can you reverse or inhibit those changes? So that's sort of what we are focusing on doing for corneal endothelial cells. And the clinical rationale is very clear. We need better cells for cell therapy application for patients. So that is one of the focus in my lab. Talking about the tissue engineering approach, and there is a strong rationale that why do we want to make a monolayer in the lab? We can just inject the cells, right? With a cell injection, there are some issues. You don't really have a control of how many cells will attach. And you also need to inject 3 times more cells, because some of the cells through trabecular meshwork, they are lost and they are even found in the liver, for example. So there are some challenges with that. So we thought that it would be great to have a thin film of hydrogel on which that can support as a physical, basically, support, right? We can grow the endothelial cells on that hydrogel film, and then we can transplant that. Now, that was the time where GELMA was basically being used And we tested Gelma and compared to other controls, hydrogels, and we found that the cells did not require any coating and they were attaching very nicely with the gelatin methacrylate based hydrogels. So, but there was a key issue. When our clinical collaborator, the Jod Mehta surgeon at Singapore Eye Research Institute, they tested that Gelma hydrogel film. It was about 150 micron thick. So they rolled it, they tried to put it in the eye, but it would break easily. Like, I mean, that was not basically good, right? The hydrogel film, if it breaks, basically you're damaging your tissue construct that you want to transplant into the patients. And that I'm speaking of a surgeon that is like well known to do these kind of surgeries in patients. So we needed to improve the strength. What we eventually could figure out that if we now generally we keep the, I mean, this audience is very well aware of the Gelma. So we generally keep the Gelma at 37 degrees centigrade. We shine light in the gels. We combine physical cross-linking that naturally happens in gelatin-based solutions, that if you cool it down to 4 degrees centigrade, it would form physical gel. And then we cross-link the Gelma in that state when it was already a physical gel. And we showed that for example, at 30% concentration, we call that Gelma Plus. The Gelma Plus had almost 5 times higher mechanical strength compared to regular Gelma, just by adding this tweak of cross-linking when it's already a physical gel. And our surgeon really liked it. He could 100%, with 100% efficiency, was able to transplant that kind of monolayer into the corneas. So that's sort of fixed one of the issue that we had. Now, because we knew that having topographies or 1 micron topographies on the surface could improve the properties, so we developed a nano imprinting method to basically pattern these 1 micron pillars on a hydrogel film. So this is an example of a dried Gelma film that has these 1 micron pillars, and that's why it's reflecting light. And indeed, it showed that we can improve the tight junction formation on this kind of graft. This is an SEM image of a monolayer at passage 3 that is grown on a patterned hydrogel film. Did some analysis showing that Z01 is indeed increased on a patterned hydrogel film. Cell density is also better. So the things were looking good. Then we transplanted this in rabbits, and we followed that for 4 months. This is an OCT figure. So basically, this is cornea. This is the hydrogel film on this side. And you can see this is at day 10 in vivo in rabbit eyes. By day 20, we could still see the hydrogel film there, right? We took that hydrogel film out. We did some pore size analysis, and it showed that the Gelma Plus film had significantly wider pores, which indicated that it's degrading. Our thought process was that Gelma is degradable. It responds to collagenase enzymes, and therefore, when we will transplant, it is exposed to aqueous humor, so it should gradually degrade. But it was not degrading at a rate that we expected, right? Now, that is a huge challenge. Why is that a challenge? Well, that's a challenge because the Gelma is there to provide a physical support. We did not intend to have Gelma for the rest of the life of the patient. We thought that eventually it will degrade, and meanwhile, the cells will secrete their own ECM, and they will eventually establish their own contacts, and they will integrate with the host cornea. But if the hydrogel does not degrade, we are not able to study that next question, whether the cells will eventually integrate with the cornea or not, right? We cannot do an experiment that is like a year long to find that out. So we needed a way to decouple the degradation rate from basically the mechanical properties, right? Obvious way to improve the degradation rate is just reduce the concentration of the hydrogel or reduce the cross-linking. But that, remember, reduces the mechanical properties, right? Which we cannot transplant. So we needed to decouple these 2 factors. And that generally is a challenge, because if you want to have a higher biodegradation rate, basically the strength will be lower, right? These 2 things are kind of intertwined. So how do we develop a gel that is strong yet has a tunable biodegradation rate? What we did is we thought, so I was doing my postdoc in Molly Shockett's lab, and I had a fascination of controlling the hydrogel properties using light. And we were collaborating with Dr. Mark DiStefano from University of Minnesota. They had developed these nice molecules, methoxynitrodibenzophuran, which responds with high efficiency to 2-photon laser. So for example, here, what I did, I took a hyaluronic acid gel and had a thiol group, which was masked by using this group. And we tested the efficiency of cleavage by exposing this to 2-photon laser, which would then cleave the bond, expose the reactive thiol, which could be linked to maleimide dye. And we showed that indeed we can actually very precisely conjugate molecules that would react to thiol where we want and when we want in a 3D space. And I thought that can we develop a cross-linker that is composed of this molecule so that after transplantation, we can use a femtosecond laser, which is, by the way, very commonly used in ophthalmology. And we use that laser to cleave that hydrogel film, right? But that would be cumbersome because doing that at that scale is much better, much faster. But if you want to degrade the whole 1 centimeter diameter film, it would be a very cumbersome process. So what we are doing in my lab funded by NIH is we are developing basically, I would say, a range of hydrogels to test their properties that would respond to an exogenous or external stimuli. One of that stimuli is NIR cleavable linker. So instead of using the previous version that responds to 2-photon light, we are collaborating with Dr. Osama at UT San Antonio. So Osama's lab has a lot of expertise in making molecules that respond to near-infrared light. So they use that as dyes and for other purposes to deliver stuff in the cells. We are using that as a cross-linker to hydrogels so that it responds to a simple lamp, basically, NIR lamp, instead of a $1 million femtosecond laser. Another version that we are doing of the hydrogel is a hydrogel that is cross-linked with a peptide sequence that responds to sortase enzyme. So sortase, again, Quality Forest Lab, for example, at Washington, has published very nice papers showing that sortase is pretty orthogonal, does not interfere with any of the processes that happens in the body, and that sortase can cleave those bonds within 30 minutes. So we thought, okay, that's great. If we can make a hydrogel that responds to sortase, that allows us to use sortase as an eye drop after transplantation of that tissue-engineered graft. So then this allows us to degrade the carrier at different days to see what time period is the most effective time period to improve the integration of the transplanted cells. So I'll switch to the other story now. The work that I did in Molly Shawkett lab on developing basically bile duct organoids. So, and generally in the field, I was more intrigued by this project because if you think of liver tissue engineering, a lot of people have been focusing on hepatocyte organoids, right? Pretty much that's what considered a liver tissue engineering. Bile duct was an afterthought, right? That you can develop stuff with bile duct organoids, and the bile ducts are important. And I guess rightly so, because hepatocyte organoids are very important for drug toxicity studies. So there was a lot of focus on developing that. I picked up that project because, I mean, I was intrigued by the bile ducts and I thought, okay, something liver related allows me to basically sort of use my hydrogel development skills and see how we can develop bile ducts. Now, just a brief background, the bile ducts, you might know that are basically are there in the liver to collect the bile acids that are produced by the hepatocytes, right? So the bile ducts eventually merge into common bile duct and they'll collect all these bile acids and take them away during the detoxification processes, right? Now, there are several diseases, and the cholangiopathies, which collectively are basically the bile duct diseases, almost 1/3 of the liver transplants in adults and almost 2/3 liver transplants in pediatric patients, right? The bile or cholangiopathies are one of the key reasons for liver transplantation itself, right? If your bile ducts are not functional, that leads to eventually cirrhotic livers that needs to be transplanted. Now, our collaborators at in Toronto, they had published a protocol for the first time developing the cholangiocyte cells from iPSCs. But during that process, they used a feeder layer that was jagged-1 positive feeder layer. Now, jagged-1 is a ligand that activates Notch signaling in these cells. So they used jagged-1 to provide Notch signaling to the hepatoblast cells. Then they would eventually differentiate to these bile duct organoids. We thought that, is there a way to replace that feeder layer with something more synthetic that we can control? Now, Notch signaling is well known to create the bile ducts in the liver. For example, this fetal study showed that liver study showed that JAGR1 is expressed, for example, at around E12.5 and only in specific locations. Now, it's very interesting to see that HES1, which is a marker of Notch signaling, is also expressed only where JAGR1 is expressed, right? And then if we give it a couple of more days, you start to see CK19, a marker of bile ducts, again, only expressed where Notch signaling was working, right? And then we start to see these bile ducts forming near these portal veins, right? So what happens to these rest of all the rest of the cells? Well, they become either hepatocytes or non-parenchymal cells. So notch signaling clearly controls which cells are becoming cholangiocytes and eventually forming bile ducts, and which cells will eventually become hepatocytes. It is so potent that hepatocyte differentiation protocols actually involve notch inhibition step, because if you activate notch signaling in hepatocytes, they de-differentiate. They'll lose their markers. Okay. So we thought that, okay, can we control this notch signaling spatially and temporally? Because it clearly happens during the developing liver. It is controlled spatially and temporally during the process. So the strategy that a postdoc in Molly's lab, excellent organic chemist, and I come up with a strategy where we would cage the jagged ligands conjugated on a hyaluronan hydrogel, and that cage would limit the interaction of the Notch2 receptor with the jagged1 protein. And the idea is that if we photo uncage this cage, that would then initiate the Notch signaling process. And because it is controlled by light, this would allow us to control where we want to activate the Notch signaling process. Right? Or on the other hand, we can directly just add jagged1, which is an active state. And if we just want Notch signaling throughout the matrix, we can activate throughout the matrix. Right? So with that strategy, we tested that linker. So she synthesized that linker that basically had a very well-known like orthonitrobenzyl moiety here, NHS ester on one side, biotin on the other side, that could bind to streptavidin. So we had a hydrogel, we conjugated protein G, and then protein G was linked with FC jagged protein, so that the jagged one is at a proper orientation instead of directly modifying the jagged one. And then it was linked with these photolinkers that had streptavidin already attached to them. So this way, jagged one was sort of masked by these bulky streptavidin molecules so that the receptors would not bind to it. And when we shine light on it, we can expose the jagged one. We tested that by using a dye which was linked with the streptavidin. And clearly we showed that if you shine light, you remove that dye. So the red areas are here in which jagged is still masked, but these darker areas are where we actually remove the streptavidin molecules from the jagged, making it sort of on state. Does it work? Yes, it indeed does. Dr. Elowitz's lab was very nice at Caltech. So I contacted them. They sent us a notch reporter cell line, which becomes fluorescent when the notch signaling is active. So we showed that within the same 96 well plate, if you cover half of the area with the mask and then expose half of the area, the cells become active, or Notch2 becomes active in cells only in those regions where you shine light. And you can see the effect actually within 8 hours, pretty much. Within 8 hours, those areas which were exposed, they would show that the notch signaling is active in those cells. Then we verified that, yes, indeed, it also works temporally in iPSC-derived cholangiocytes, where if you don't expose those wells, the cells will not differentiate to cholangiocytes. But if you expose to light, they will differentiate. They will express SOX9 as well as other markers such as SOX9, CK19, and CFTR at both gene level and protein level. So that was showing to us that indeed this strategy works. You can sort of control the notch signaling through light. But we wanted to do this in 3D, right? So in 3D cultures. So we developed a hydrogel. Let me see how I'm doing on time. Okay. So we developed a hydrogel in which basically we modified the chemistry a bit so that it's faster cross-linking, and we also conjugated the jagged1 protein in the hydrogels. And we showed that if we encapsulate the cholangiocytes in the hydrogels without any laminin, the cells do not really grow in it, which was unfortunate. But as we start to increase the amount of laminin in it, eventually the cells will start to grow in this HA laminin hydrogels, which the growth rate was pretty similar to a metrogel. So they were growing these organoid-like structures. That was intriguing because the amount of laminin, let me point out. So 0.1% laminin is actually a lot of laminin. This is 1 milligram per ml laminin, way more than what people would generally use in a hydrogel to increase, for example, cell adhesion, right? So we were thinking that maybe something else is happening. It's not just that cell have laminin, they can adhere to laminin, probably something else is going on. And one of that thing was, I was intrigued by looking at the viscoelasticity or the stress relaxation of the matrix. So we analyzed that if without laminin, the hydrogel is not very stress relaxing. But as you increase the amount of laminin, eventually the stress relaxation rate of the hydrogel is similar to liver as well as to matrigel, right? And this is the composition in which the organoids were growing much faster, right? And is it actually stress relaxation? Well, we took another hydrogel in which we added more HA and crosslinks and had the same amount of laminin, 0.3% laminin, but the organoids did not grow in this gel. So it indicated to us that it's not just that the laminin is present, it's actually that the hydrogel needs to be more viscoelastic to allow these cells to sort of remodel their environment and grow as an organoid. We further showed that stress relaxation allows the activation of YAP signaling in the 3D culture. And we looked at the expression of these four YAP target genes, which were upregulated in a stress relaxing matrix, but not in the same laminin composition matrix that was slow relaxing. And if we inhibit the YAP signaling through a small molecule called verteporfin, the organoids do not grow, even though it's a stress relaxing microenvironment. That again showed that the YAP signaling is involved. It may not be the only factor, but it is involved in improving the organoid growth in a 3D cell culture matrix. So what happens if you activate notch signaling or in these cells? So we modified the hydrogel and conjugated jagged one signals in the hydrogel. We verified that in these, it basically upregulates the HEA1 and HES1 expression, the well-known notch target genes. By day 7, we could start to see that the cells, instead of just staying as organoids, they have started to sort of contact with each other and form these bile duct structures. By day 14, pretty much all the cells had transformed into these kind of well-connected bile duct like of network in a 3D culture. If you zoom in, like at higher magnification image, you see that the cells are aligned at these walls, and they clearly have lumen. And this is just a Z-stack showing a 3D architecture of these forming bile ducts in the culture. But again, nothing happens in a stiffer, or not stiffer, less viscoelastic matrix, even if you have the same amount of laminin and jagged one there. So clearly, again, further indicates that, again, the jagged one alone or the viscoelasticity alone is not enough. You need appropriate combination of both of these two factors to improve this. Now, in this hydrogel, we are still improving viscoelasticity by laminin, right? But laminin still provides other signals. Like laminin is well known to basically improve the organoid growth, but it's not just the mechanical properties. So we wanted to figure out a more sort of defined hydrogel in which we can decouple and improve the stress relaxation or tune the stress relaxation without adding biocomponent factors. So I reviewed a bunch of chemistries that are called dynamic covalent bonding chemistries used in the hydrogels. And we know that pretty much all the tissues have some sort of viscoelasticity, probably except bone. And on this spectrum, the permanent cross-linking hydrogels, such as we used previously, or for example, methacrylate-based cross-linking, generally have much more stability, but usually the viscoelasticity of the hydrogel is lower. On the other hand, dynamic covalent cross-linking provides you a range of cross-linking, a range of viscoelastic microenvironments. Non-covalent bonding is the best, which is often seen in extracellular matrix, but usually the hydrogels are not very stable for 3D cell culture if you're purely relying on a non-covalent interactions. So out of those chemistries, one chemistry really stood out to me. It's basically a molecule called lipoic acid, which contains a disulfide bond in this 1,2-diethylene ring. Now, this stood out because disulfide exchange is a very well-known mechanism. It's a very dynamic process that happens all the time in the body in different protein interactions or ECMs. So we thought that can we conjugate lipoic acid or this diethylene to a matrix and then somehow get a hydrogel out of this? Now, even though disulfide bonding is very well known, but there are not many hydrogel systems because it's very difficult to work with disulfides. Right? They get oxidized very easily. It's difficult to basically control the process. So what's special about this? Special about this is that this is a strained ring, which means that as soon as you shine UV light on it, it opens up and it's sort of freshly generated thiols react with the other thiols nearby, and they will form these disulfide crosslinks. So those sulfides are kind of naturally protected or naturally masked dithiols unless you provide some energy, which is the light energy. Now, it also does not require any photoinitiator, right? Because you are just purely relying on that, the ability of these rings to open up when supplied with the energy. So does it form a hydrogel? Yes, indeed it does. As if we looked at the rheology basically of these hydrogels, so as soon as you shine light, within seconds, the hydrogel goes from solution state to a gel state. And we tested this with gelatin diethylene hydrogel from 2.5 to 10%. And indeed, it forms a hydrogel. Frequency sweeps show that it remains pretty stable. So it gives a very nice hydrogel system that is composed of disulfide crosslinks. The hydrogel, we were worried that because these are very dynamic crosslinks, so the hydrogels may not be stable for long-term studies, which are often required for organoid culture. But no, these hydrogels are pretty stable. For example, up to 30 days, they actually maintain their weight. There is indeed some swelling, but the hydrogels maintain their shape. You can tune the mechanical properties. The hydrogels, as we expected, are much more less relaxing compared to the permanent covalently bonded hydrogels. We showed that indeed the growth rate, for example, in the dynamic cross-linked hydrogels, even though both are gelatin-based hydrogels, is higher compared to the GELMA, which has more permanent covalent bonds. We tested this using cholangiocarcinoma cells, which form tumors which were more representative of the tumoroids that were seen in patient tumor cells or patient tumors. The tumor size was also bigger, so they were growing better basically in the gel-DT hydrogels. Okay, because it has disulfide-based cross-linking, and we know that a lot of the proteins or growth factors have cysteines, so we thought that they should be able to interact within the matrix. So the matrix should be able to retain those disulfides. And so what Cody did, that Cody mixed the EGF, which has, I think, 9 or 5 cysteines. They mixed this with the hydrogels, the gelatin diethylene solution, cross-linked it, and showed that indeed it really retains the proteins very nicely. And the release is also much, much slower. So for those applications in which you want hydrogel to retain basically or release it very slowly, the proteins, this hydrogel would serve much better in those scenarios. And indeed, the retained hydrogel, retained EGF was also active because we tested it by using the cell culture where the EGF is supposed to promote the growth rate of those cells. Now, another benefit that then we thought, okay, can we tune the stress relaxation post-gelation? Right? During the cell culture process, we showed that if you add exogenous compounds which contain thiols, for example, the 4-arm PEG-SH here, it basically can interact the already cross-linked hydrogel and can increase the viscoelasticity while keeping the stiffness same in all these conditions. For example, here, the SAD treated the hydrogel with the PEG-4SH for different amounts of time and showed that the stress relaxation amount or rate depends on the time the hydrogel is exposed to PEG-4SH. And this is something can be done in the presence of the cells. So you can really dynamically tune the stress relaxation environment as you are doing the 3D cell culture. Now that got us wondering that if the crosslinks can react with exogenous thiols, what if instead of light, because in some scenarios, light is not accessible, right? You're injecting a hydrogel in ischemic stroke model. You may not be able to provide light to cross-link the gel there. So we thought, can we benefit from this hydrogel in those kind of scenarios where we cannot shine light to activate this? So we thought if we activate these compounds and mix it simply with PEG-SH of various structures and molecular weights, what happens? Well, I had a grad and undergrad student. They analyzed, I think, 81 different formulations. They took PEG of 3 different structures. I'm not exposing here since this is being recorded, and of 3 different molecular weights. And they also varied the amount of basically molar ratios of PEG-SH to diethylenes and also the different concentrations of diethylene. So a lot of factors. We wanted to figure out there got to be some structures which allows these diethylenes to open up without even using light. And indeed they do. They looked at gelation speed. The gelation speed here, basically the color represents how fast these gels form a gel. Some of these, actually almost half of these gels form instant gelation in some of these structures. Some gels did not form any gel. So really the PEG structure was controlling the rate of gelation as well as the stability. Only Some of these gels were stable for more than 24 hours, and now my students have tested, they remain stable for up to weeks. But some gels just dissolve within a day. So basically, PEG here controls how stable the gels are and how fast they will form a gel. Also, we tested that if you apply gel DT on a corneal stromal wound model, that is one of our interests of basically applying this gel. The gel DT stays there very nicely in a stromal wound model. And this was basically put on a shaker, the eye globes, to basically ensure that we are applying some shear forces to these hydrogels. But it remains very nicely. However, the gel SH does not have that much strong bioadhesion to wounds, which again might be dependent on, because gel DT was directly cross-linked on the cornea. So the thiols, as soon as they open up, they sort of react with the native proteins as well in the wound area. This is why they might be able to have a strong bioadhesion component there. In the last couple of slides, 3 slides, I'll just mention another biomaterial that we have been recently started work on. And what really motivated us to work on this biomaterial was that a lot of the pathogenesis is basically the inflammation and oxidative stress is implicated in a lot of the diseases or pathogenesis. For example, I'm focusing on ophthalmology, where pretty much dry eye, diabetic retinopathy, the degeneration of retinal ganglion cells, they have all been linked with this radiation that the eye is constantly exposed to, and that generates reactive oxygen species, inflammation, and eventually leads to degeneration in that case, right? So we thought that, can we develop an injectable gel that naturally has a well-known ability to reduce inflammation wherever we inject? And several labs, for example, Segura Lab, have shown that if you can reduce inflammation, the impact of growth factors is amplified. Like, they can work much better in a non-inflammatory microenvironment. So we stumbled upon this material or polysaccharide called fucoidin. It's an FDA-approved food supplement that has well-known antioxidant and anti-inflammatory properties. In some ways, this is similar to a heparin molecule, which is basically a polysaccharide, and it has some sulfates groups attached to it, similar to a heparin. Now, in literature, you will not see a lot of hydrogel-based systems, but people have mixed this as a component in other hydrogels to leverage from its properties. In this case, actually, Evelyn James' lab, they made vascular grafts and grafted fucoidin on the surface to take benefit of the anticoagulant properties of this. And they indeed showed that in a rabbit model, the vascular grafts remain patent for much longer if they have been modified with the fucoidin. In another study, they showed that insulin secreting cells, which are or these pancreatic beta cells, they function much better in an alginate hydrogel if it is also mixed with fucoidin. But we were purely interested in making a fucoidin-based hydrogel instead of adding fucoidin as a soluble component to other hydrogel systems. So my students have actually done a lot of studies, really a team effort, where they modified fucoidin, purified it, modified it, and cross-linked it using either hydrazone chemistry or imine cross-linking chemistry. Again, both are dynamic covalent chemistries because of our sort of love affair with them for 3D cell culture applications, as well as for injectability that these hydrogels provide. These hydrogels, again, I'm just showing very selected data in these 2 slides, provide a lot of tunability in gelation time. You can make them gel instantaneously, or you can make them gel maybe after 10 minutes, depending on what kind of concentration and molar ratios you're using. These gels are stress relaxing, just again because of the dynamic chemistry. Their gel mass remains pretty stable. They show nice injectability as well as the bioadhesion with the structures. Lastly, very exciting, what we are seeing is that indeed the fucoidin hydrogels, both in oxidized form as well as in the hydrogel form, they can remove the inflammatory signals and they can improve the pro-regenerative signals, which is what keeps us exciting to continue to work. And the student have already injected neurons, which are mixed with an imine cross-linked hydrogel, and showed that after injection, even though these hydrogels require different amount of force for injection, but pretty much in all 3 formulations, the neurons survived very well after 7 days of injection through these hydrogels. So I think this really provides us a framework to continue to work on injecting these cells, for example, the retinal ganglion cells, or using these cells wherever there is inflammation involved. And there are so many opportunities basically with that. With that, I'll end my talk. I'll thank my lab. This is the most recent pictures, and also with my kids. And my collaborators, we have a lot of collaboration within USA and also across in Singapore, as well as in Germany now, and funding sources. So thank you very much for listening.
Thank you, Rizwan. Thank you for the nice talk. Questions?
So thank you very much for your talk. Great stuff you're doing. So I had one question regarding your endothelial cells at the beginning of your talk, and I was wondering, like endothelial cells, many times in the lab, one thing they like is shear stress. And
Yeah.
so in the eye, I was wondering what kind of shear stress there is in the eye, or and is that really related also to how specific those cells are? And after, did you ever try to grow them with shear stress?
Oh, you're awesome question. You're not the first one who actually asked this question. That was not something I thought a lot about as a grad student when I was working with these cells or initially. But the more I've looked at the effect of shear forces on the cells, and then the more I looked at the rate of aqueous humor flow, like there is a continuous flow of aqueous humor. So these cells throughout their life, they have been exposed to a liquid aqueous humor which is flowing at a particular rate, right? Which is applying some sort of shear forces, right, on these cells. Unfortunately, so far there is no study that has done any analysis. Actually, even the basic questions of in mechanical transduction is just not well known in endothelial cells, just because it's very difficult to isolate these cells, with work these cells. These cells are very difficult to grow. Even if you can get 3 doublings, like that's like the best already. So that is what limits. Now you can work with immortalized cells, but their history is very different because they have already been exposed to so many different environments. By the time you get a vial from someone, like there's only one I think company in Germany who sells this. So briefly, yes, I think my hypothesis is that yes, shear force will have some effect on these cells, but currently there is nothing known. So what we are thinking of with our collaborators, that maybe we can build a system in which we can take aqueous humor, because Singapore group, they usually sends us aqueous humor isolated from rabbits, and maybe we can flow that and see if we can improve the maturity of these cells.
All right, thank you.
More questions? No?
So then after also with your organoids or your cholangiocyte organoids or bile duct organoids, so as you were showing them, I was thinking like, okay, are they forming those bile ducts, which you have shown? But so what are the characteristic or how big is that bile duct network that you're forming with those organoids? Or like morphologically, like what is the range of size of ducts?
Yeah.
Or...
So you're talking about this, right?
Yes.
So I was working. Good question. I never tried in a bigger gel.
Mhm.
I was working in, let me remember, maybe 20 microliter gel size. And we could see this network throughout, like most of the gel would have this kind of network, right? But again, thinking of ARPA and the size of the things, maybe we should try if we can get something similar in on a bigger scale. But so far we haven't tried. But there is another issue actually. The not issue as in just tangent to this, that this activates NOT signaling in a gel, and you can form bile ducts, right? But I started thinking of liver, and let me actually show you a very nice 3D reconstruction of an organoid from. Okay. So this is a reconstruction of a well-known group of an organoid, which they show these bile canaliculi in these organoids, and these big yellow sort of balls here is the accumulation of bile acids within these organoids. And we think that one of the and well known again that the hepatocyte cells don't really function for a very long time in culture. They may grow better in spheroid, but again, spheroids also don't really function for a very long time. And we think that one of the reasons is that there is no way for these bile acids to get out of these organoids. They kind of stay there, and that leads to eventually the degeneration or de-differentiation of the hepatocytes, and that's why they lose their function. So even if we are able to grow bile duct network in a hydrogel, we have to think of ways that we can form those bile duct networks in the presence of hepatocytes as well, right? or find a way where we sort of convert some hepatocytes towards the cholangiocyte lineage. Eventually, they form bile ducts. Well, one obvious way is, well, let's activate notch signaling. But the problem is that, as I said, like notch signaling, if you activate in hepatocytes, they differentiate. So you really need a spatial way of activating notch signaling within a cell culture system, so that only in defined regions you activate the notch signaling and make these bile duct networks. Actually, we are doing something very interesting with Leonardo Morsat, who you might know. So we are doing some cell engineering approaches to see if we can maybe leverage some synthetic kind of receptors and combine them to get bile ducts in these livers. But we'll see.
Okay,
Yeah.
and then did you look? Did you do like cross section or like some H&E staining of your tubes to really see like what because it looks like there is a lumen. So after my question is really, do you think those ducts could really drain bile?
Yeah. To know, yeah, they have lumen clearly. We did a reconstruction, and yes, whether they will drain, I guess you need to combine them in some sort of organ on chip system where you have an input and output where you can flow something and see how functional they are. Not my expertise. We haven't done that, so maybe that'll be a great area if we can do something on that. The other thing is then, yeah, but we have seen their function. We expose them to the rhodamine dye, the fluorescent dye, and they will transport that dye. So that's part of the paper. They are functional, but in terms of like an input and output and whether they can take, there are actually two papers in Nature Communications from Japanese groups. I think those were the first paper that I see where rationally they put in cholangiocytes and hepatocytes, and they showed a connection of bile canaliculi of hepatocytes with the bile duct cells, and that was very, very interesting to see. People have been thinking of combining these two things together. Yeah.
I have a simple question about your retinal regeneration.
Yeah.
There's some gaps. You put the cells. Are you doing in culture inside the animal, and what happens to extra cells that don't attach? Can you explain a little?
Oh, you mean for the retinal cell delivery? So we have been working with the collaborator, Dr. Kevin Park. He has a lot of expertise in basically retinal regeneration and looking at the molecular pathways of why these cells die, or what happens, or why optic nerve degeneration happens. What we are thinking, there are two approaches in terms of cells. We can grow retinal organoids and isolate RGCs, but it takes a lot of time, like over several months to grow retinal organoids. And then it's also not easy to isolate RGCs. They're sort of inside the organoid sometime. And so it's very difficult. The other approach that we are thinking is just using what my student currently she's doing is we are just following a protocol published by another lab, which instead of using organoids, just differentiate iPSCs to neural progenitor cells and then directs neural progenitor cells towards the RGC-like fate. Now, we're not sure how mature or how functional that will be, but they have shown good results with that. And currently, my student is basically working on that. And
You show a membrane,
yeah,
with some patches. Can you show that membrane again? And you were injecting some Dimitri.
That was in the first part with endothelial.
Dimitri membrane, really from the beginning.
Okay. So this is the Gelma film.
No, there is a membrane, Demicus or some retinal.
Tight junction.
Okay, let me see.
No, one above. There's, you say, yeah, this one. So there's gaps, endothelial dystrophy.
Yeah.
You bring the cells, and the cells find those holes and attach. Is that how it works?
Oh, no, this does not. No, sorry for the confusion. This does not really cause holes. This black, you might think of that. No, this doesn't really cause holes. What happens, actually, no one really knows why this happens. People have theories of how the ECM changes from a flat surface to a surface which contains these microstructures over time. It obviously doesn't happen overnight. Like it happens over weeks and probably months and even years. But the surface, this is an SEM of a posterior corneal surface. So the ECM, which was supposed to be a flat and then have a nice endothelium layer, instead of that, the surface now has these sort of micropillars. Right? It's something you would use lithography to make micropillars. And due to these, as they start to appear on the surface, they eventually cause the degeneration of the cells. In fact, I didn't show here, I made a lithography-based model that would recapitulate these sizes in vitro, because we were intrigued by Japanese groups when they were injecting cells, that how can cells, if the patient already has these Gauté structures, right? How could cell injection therapy be successful, right? Because you have to think about these Gauté structures. So intrigued by that, we made a model in which the surface of polystyrene had these various profiles of Gauté structures. We did a simulated sort of cell injection, and we clearly showed that cells are not able to form a monolayer if the surface already has Gauté structures. And those studies have been replicated by other groups as well. So then I asked the Japanese group that, and my collaborators also, that how are you able? And they said that in Japanese patients, 90% of the cases of endothelial dystrophy is not because of Gauté, which is unlike the rest of the population in the world. So the most of the Japanese patients, there are genetic causes or there are some other comorbidities that basically lead to this. So the patients will have endothelium dysfunction, but the reason is not Gauté. But in the rest of the world, Gauté is the main reason. So for that, you have to actually remove the Gauté using probably laser that's already in use in clinics, and to make the surface flat, and then you can do cell injection therapy. That's the only way it could work.
So yeah, that's what's related to that actually also. So here, your film, if you try to implant it, so to replace that endothelial cell layer, it's really a 5 micrometer thick layer where you have a flap or a gel that is 150 microns. So I wonder how, how your 5 micron layer is going to really go through and really replace the existing layer where you have,
Wonderful question. For 2 reasons. I asked Jod, like a clinical surgeon or corneal surgeon, that can we not put it up other way around, right? Where the cells are facing the cornea, right? And the gel is on the other side, so that we don't have to worry when it will degrade. Whenever it will degrade, it will just degrade, right? And he basically said that we had tried that already. What happens that when the cells are already formed a monolayer on a hydrogel, they are polarized in a way that they cannot function if you put it the other way around. Because there's a direction here. These cells are supposed to maintain the hydration in the cornea, right? So they have to pump ions in a certain direction, right? Which cannot happen. Now, to the other question, yes, the cells are 5 micron only, right? And the hydrogel is around 100 micron. So at some point, when the hydrogel is gone, what happens to that monolayer? Like, will they be able to establish a connection there? Well, that's a question we are studying in the R01. But just to briefly tell, we think that when the hydrogel film eventually degrades, the stability of the monolayer will depend on how much extracellular matrix they have secreted under them and how tight the Z01 junctions are, or cell-cell junctions are. These 2 things will collectively define if, after the material is gone, whether they will stay as a monolayer or will they disintegrate into fragments, right? To do that, we are actually looking at metabolic labeling of ECM secretion in these cells, and then quantify how much secretion these cells have and what kind of surface topographies can improve the secretion of those proteins. Yeah.
Yeah. There's a question, one more question, last question. So is the transparency of the biodegradable hydrogel is important for the regeneration of corneal epithelial tissue, while regeneration occurs in a short time?
Yes. Transparency is important in a sense that, for example, this gel, if we see, even though it may look that it's not transparent, but that's because it's dry and it is reflecting light in a certain way that it looks not transparent. But once we submerged it in the media, this is a pretty transparent. You can't even tell basically where the hydrogel film is. Transparency is important in a sense that, well, if it's not a transparent gel, for the period where the gel is still there, the patient will not be able to see, obviously, when this is transplanted, right? So ideally, we would want to have a gel that is transparent in that condition. Thank you.